Hello everyone, welcome to today's live broadcast, Early Days of the NIH Brain Initiative. I am Judy O'Rourke of LabRoots and I'll be your moderator for today's event. We have a few important announcements before we begin. This presentation has been approved for a Category 1 Continuing Medical Education credit by Loma Linda University. If you're interested in obtaining CMEs after attending this presentation, please click on the Get Your Free CME CE Credits button in the bottom left corner of the event screen. This webcast is designed to be interactive and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Dr. Walter Koroshetz. Dr. Koroshetz was named Acting Director of NINS in October 2014. Prior to this appointment, he served as Deputy Director of NINS since January 2007, where he worked with the NINS Director in Program Planning and Budgeting and overseeing Institute scientific and administrative functions. Before joining NINS, Dr. Koroshetz served as Vice Chair of the Neurology Service and Director of Stroke and Neurointensive Care Services at Massachusetts General Hospital, MGH. He was also a professor of neurology at Harvard Medical School and has led neurology resident training at MGH since 1990. A native of Brooklyn, New York, Dr. Koroshetz graduated from Georgetown University and received his medical degree from the University of Chicago. He trained in internal medicine at the University of Chicago and Massachusetts General Hospital. Dr. Koroshetz trained in neurology at MGH, after which he did postdoctoral studies in cellular neurophysiology at MGH and the Harvard Neurobiology Department. He joined the neurology staff, first in the Huntingtons Disease unit and then in the Stroke and Neurointensive Care Service. I will now turn it over to Dr. Koroshetz for his presentation.
Hello. This is Dr. Walter Koroshetz, and it's a great pleasure to talk to you. It's evening here. Very relaxed time to communicate to you a very important topic, the National Institutes of Health Brain Initiative. As Judy mentioned, I am the Acting Director of the National Institute of Neurological Disorders and Stroke, one of many institutes at NIH that fund and conduct research in neuroscience. The Brain Initiative is a unique initiative on the part of NIH, as it did not come from NIH. Originally, it came from the President of the United States. The White House recognized the need for advancing neuroscience research and put forth this initiative, which is to advance brain research through advancing innovative neurotechnologies. So it's a very neurotechnology-driven project and unique to the NIH at this point. The NINDS, as I mentioned, is one of many institutes that funds neuroscience research, and in fact, neuroscience research receives the most funding of any general category at NIH. Approximately $5.4 billion per year are coded as neuroscience research. This research is done throughout multiple institutes. Our institute is 100% neuroscience research. There are other institutes which are heavily neuroscience research, such as the National Institute for Mental Health, the National Institute of Drug Abuse, and then other institutes have varying degrees of neuroscience portfolios, such as the National Institute of Aging, which is charged with developing better therapies for Alzheimer's disease, but also aging research in general. National Institute of Child Health and Development, for instance, funds research and development of the nervous system. And these institutes, although they work individually with their own priorities and portfolios, come together in what's called the Neuroscience Blueprint at NIH. And that group is very much invested in the Brain Initiative, and much of the support for the Brain Initiative has come through the Blueprint, in addition to additional funds from congressional budget to NIH for the purpose of advancing the NIH Brain Initiative. I imagine that many of you who are listening tonight or this morning or wherever it is, wherever you are around the world, recognize the draw that understanding the brain has for millions and millions of people around the world. The brain is inherently interesting because it is really the physical basis of who we are as humans, who we are as individuals. So it can gather the imagination and the interest and curiosity. As an example of that, the media has certainly taken advantage of this interest in terms of bringing out to the public the messages about the brain and the new brain science. In the United States, of those going into PhD programs, the largest percentage of PhD students are entering neuroscience, and this is growing compared to the plateau that is seen in many of the other science areas. So neuroscience is a growing field, and the Brain Initiative is really trying to develop the technologies that can arm these new investigators coming into the field to make greater advances in the science of how the brain functions. Now, as I mentioned, the brain is inherently interesting to many millions of people around the world, but there is a very real reason why it should have interest to those interested in the public health as well. And that's because if one looks at daily adjusted life years of disability, disorders of the brain are the leading cause of disability in the U.S. and actually in most developed countries, and is growing around the world, even in the developing countries. Neuropsychiatric disorders and neurological disorders combined are the leading cause of disability, followed by cardiovascular and circulatory disorders, and those actually include stroke, which in our vernacular is considered a neurological disorder. Neoplasms come third, musculoskeletal disorders fourth, and many of the musculoskeletal disorders are characterized by pain as their main feature, and those could also be considered coming under the rubric of neural disorders. So I think there's a huge burden of illness that people are suffering with due to disorders of the brain and the nervous system. And these disorders, the symptoms, the problems that they cause, are related to abnormal functioning of neural circuits. So let me explain that for a second. So for instance, in the condition of a stroke, there's an area of brain that is damaged. But what matters to the person is not necessarily the area that's damaged, but the symptoms, the things that they can no longer do because of that brain damage. And that's related to the circuit function in the brain. When the damage occurs, there's often maximal disability. But over time, the circuits become better able to function. And most patients with stroke who survive will develop improved function over time. And that's because the circuits in the brain are rewiring and function is improving. In those patients in whom function does not improve, the circuit changes are not translating into improved function. So the point here is that it's the circuit behavior, the circuit activity that's linked to the behavior. The circuit activity and the malfunction of that circuit activity is what is most closely related to a patient's deficits and the burden of illness. So at the center of trying to help patients is understanding the circuits of the brain, how they function in normal individuals, and what is the nature of the dysfunction in those people with neurologic or mental health or even substance abuse disorders. So the point here is that circuit activity is key, and that disordered neural circuits are responsible for a large burden of illness around the world. Not only are these disorders disabling in an individual person, but one of the reasons they account for so much disability around the world is because they often start at a younger age, and so the disability is lifelong, as opposed to conditions which occur later in life. This is true certainly for all the developmental disorders, and we consider things like autism, mental retardation, multiple different genetic disorders that affect development of the brain as this group of disorders that affect and cause disability throughout the life of an individual. And that's one of the reasons these brain disorders account for such a large percentage of disability. If one looks at Alzheimer's disease and these other diseases of aging which lead to dementia, it's another reason why we are very worried about the burden of illness due to neurological disorders. As the populations age in the United States and the other developed countries, Alzheimer's disease and dementia become a much, much larger problem and actually put serious strains on the ability to care for the affected individuals as the numbers increase. And again, the example that I used before with stroke, I want to come back to, and that is that when one looks inside the brain of an elderly individual, there are signs of Alzheimer's disease, but there's a disparity between the degree of involvement and the patient's deficits. And that's because the biology that's ongoing inside the brain, the Alzheimer's biology, is affected. The effect on the patient is mediated by how that affects changes in brain circuits. And for reasons we really don't understand, some individuals seem to be resilient and their circuits function better than others, leading to less severe problems with cognition and memory. The point again being that the Alzheimer's biology is causing trouble to patients because of the way it affects brain circuit function, whether it be memory or planning, speech. Again, the circuits are at central attention. And the reason I'm going to concentrate on the circuits because the Brain Initiative is focusing on those tools which will allow us to monitor, interrogate, and modulate neural circuits. So to summarize, brain disorders, both neurodevelopmental and neurodegenerative, we think will be the most disabling and most costly of the chronic diseases. They will be in the 21st century what infectious disease was in the 20th century. And the key issue is brain circuit function. And we have to confess that we do not know enough about how brain circuits function and how they dysfunction to cause disability for persons with neurological, mental, and substance abuse disorders. So we can examine someone for their ability to remember but we can't actually see those memory circuits in action and know what is the deficit in those persons who have trouble. Why certain persons with stroke develop incredible ability to recover function by rewiring brain circuits after injury, and why others can't. While some elderly individuals, even with significant evidence of Alzheimer's biology in the brain at death, have been functioning at a high level where others have been exceedingly disabled. What we need to be able to do is to examine and see these circuits in action, be able to monitor them over time, be able to see the circuit changes that occur years before the patient experiences disability, and then to understand how to correct these circuit disorders to restore function in the individual who's affected. Our problem is that we do not have the tools to be able to examine and monitor circuits of the brain in real time. And that is the main issue behind the Brain Initiative. It is a tool development project, and we're brought to this very prescient concept by Freeman Dyson that new directions in science are launched by new tools much more often than by new concepts. The effect of a concept revolution is to explain old things in new ways. The effect of a tool-driven revolution is to discover new things that have yet to be explained. And the purpose of the Brain Initiative is to fire up a tool-driven revolution that will give us the tools to be able to interrogate neural circuits and modulate them for health. And we believe that this is a point in time where this endeavor has a great chance of success. And that is because progress in neuroscience and technology is yielding new insights into brain structure and function. So shown here is a picture of an MRI scan using a technology called diffusion-weighted imaging to be able to map out the connections that occur between brain regions, seen here in blue and red. Prior to this ability, it was impossible to see any definition of structure in the white matter, which is the region of the brain, actually the largest region in the human brain, in which there are axons connecting the different brain regions, the actual structure behind the circuits. But MRI technology in the last 30 years has advanced to the point where this is now possible. It was never possible before. Indeed, this was a breakthrough in structural connectomics, the connections between the circuits in the brain. But there are also amazing new technologies in optics, genetics, and the combination of optics and genetics, nanotechnology, informatics, to help us design new tools that can interrogate the nervous system at the speed of light, the speed of thought, to be able to capture neural activity as it is occurring in behaving animals, with the hope that this can be translated to human studies in the near future. So it's an exciting time where technologies that have come from basic science discoveries in creatures called archaea, A-R-C-H-A-I-E, who are too primitive to be called bacteria, but have channels that activate when light is shown upon them. Taking the gene from these primitive organisms and introducing it with promoters into transgenic animals or with virus loaded genes into the brain, these channels can be then expressed in the membranes of neurons, and light can then activate these neurons in a very specific manner. This is light years away from the more crude technology of activating neurons, which is to send an electrode into the brain and then turn current on, which has dramatic effects, but very hard to understand exactly what that very crude wire conducting electricity is doing. Optogenetic techniques allow very precise activation or inactivation of particular neurons in the brain, an advance that has already revolutionized how people just do science and led to amazing, better understanding of neural circuits. What's next is a good question when it comes to neurotechnology. As a neurologist who first came into medicine in the late 70s, I was introduced to the CT scan to look at brain structure. Seen here is a 1974 picture of the brain. You can see some white around the edges where the skull is, and a little bit less bright in the cortex, and a little darker in the white matter, and then a very dark area in the ventricles, which are filled with CSF fluid. And this was amazing to be able to see the brain like this in 1975. But it's laughable now because the details are lost of what the brain really looks like. Compare this to high-field MRI scanning, seen here on the right side of this image, and the detail is quite spectacular. So this is a period of about 35 years. Where will technology take us next is the question for the Brain Initiative. And there is every expectation, because we're spoiled, that new technologies will give us much greater insights into brain structure than ever before. But the Brain Initiative hopes to go past structure and to actually see brain activity, brain function in real time as well. And that's what the President put out in April 2nd of 2013, called it the Next Great American Project. Some people call it a moonshot into the brain, comparing it to President Kennedy's challenge to go to the moon. This is a challenge to unlock the brain by, as the President said, giving scientists the tools they need to get a dynamic picture of the brain in action, to better understand how we think, how we learn, how we remember. And I agree that that knowledge will be transformative. The Brain Initiative is much bigger than NIH. It involves other federal agencies, such as the Defense Department's Advanced Research Program, the National Science Foundation, and the Food and Drug Administration. It also includes private foundations, such as the Howard Hughes Medical Institute, the Allen Institute for Brain Science, the Kavli Foundation, and the Simons Foundation. Each of these groups have a slightly different approach to the Brain Initiative, but we are working together among the federal agencies and the private foundations to leverage the investments and maximize the output. The NIH Brain Initiative is built upon a Brain 2025 scientific vision that was put together by the advisory committee for the Brain Initiative to the director, Francis Collins, the director of NIH. That group held community consultations in many different areas around the country on particular topics that are relevant to the Brain Initiative. I would encourage all of you interested in this area to read this document, which is available on the Brain Initiative website at NIH, because it laid out a vision for how the goals of understanding, monitoring, and modulating brain circuits are within reach with a significant investment of effort, funds, and time over the next 10 to 12 years. The focus, as I hinted many, many times previously in this talk, is to map the circuits of the brain, to measure the fluctuating patterns of electrical and chemical activity flowing within these circuits, to understand how their interplay creates our unique cognitive and behavioral capabilities. And I might add, to use this knowledge to understand diseases of the nervous system. I mentioned this when I first began speaking, that the disability that patients with neuro, mental, and substance abuse disorders suffer is a direct result of the disordered brain circuits, because it's brain circuit function that is translated into motion, thought, speech, personality, everything that we are. To understand them, we need to be able to see the circuits in action, so we can understand how the brain forms memories, for instance, to determine how to therapeutically modulate brain activity. One example is the improvement of motor control in Parkinson's disease, which has already been done using the very crude wire and electrical current injections that I mentioned earlier. Quite a dramatic example of how injecting current into a particular area of the brain can turn someone who's immobile with their Parkinson's disease into someone who's dancing in the room. It's an example of how modulation of brain circuits, how powerful that can be. We need to understand the circuit dysfunction and how to modulate it for benefit in the other disorders, not only neurological disorders, but the psychiatric disorders. The neurologic disorders have known pathologies that we can try to address, but for the mental health disorders and the substance abuse disorders, there is no known pathology. It's purely a circuit dysfunction. So in those disorders, understanding brain circuits is critical. The goal is to see this brain activity in the precision of individual circuits and at the speed of thought. The Brain 2025 roadmap gave us 7 high priority research areas, which I'd like to review for you now. The first of these is called Discovering Diversity. If one thinks of the brain as a computer, and it is a computer, it is the most amazing, powerful computer, more powerful, many times more powerful than anything that man has been able to construct. But to understand a computer, you need to understand its components, and that is what Discovering Diversity is about, to identify and provide experimental access to the different brain cell types. These are the cells that compose the circuits. So these are the elements of the circuit itself. We saw in that diffusion MRI the connections between different brain areas, different brain regions in red and blue. Those were the axons coming from particular cell types. And we need to understand the different types to characterize them and to determine their role in health and disease. The brain computer is a very complex one at that. The circuits occur at different levels. They occur on a microscopic level, where one cell is connected to a nearby cell. or a cell a few millimeters away. But then there are connections with areas of the brain that are very far away from that beginning nerve cell. In addition, there are multiple networks within the brain that act in concert with each other. So there are really very many different levels of brain circuit function and activity that are ongoing continuously in our brains. And so we need to understand the circuit diagrams at these multiple different levels that vary in resolution from the synapses that connect one cell to the other, to the network that occurs in a nucleus, to the network that occurs among many nuclei in the brain, to the entire brain itself. And that is what we call mapping the brain circuits at multiple scales. Third, which is mentioned many times, that we need to see the brain in action. The brain is dynamic. The activity is changing continuously. A static picture of the brain is an incomplete picture. So we need techniques that can be applied to improve our methods for large-scale monitoring of neural activity as it is occurring continuously. We have a technique called functional magnetic resonance imaging, which sees the changes in blood flow that occur in brain areas as they're activated. But the brain activity is occurring on a millisecond basis. These changes in blood flow are occurring on a matter of seconds. So there's a huge delay between the brain activity and the blood flow change. And so this technique has severe temporal resolution trouble that we need to get better techniques to get better temporal resolution. Fourth is demonstrating causality. So one can look at brain activity in many ways, and one can correlate it with behavior. But to actually know that a certain brain activity is actually causing the behavior takes another level. It takes the ability to intervene in the brain to make a change in that neural circuit to see what the change in behavior really is. And it's only by linking an intervention to a change in brain circuit function and then to behavior that we can really say we understand what causes that behavior. So demonstrating causality is closely linked to being able to intervene, to turn on nuclei or turn off nuclei, as mentioned, say, with optogenetics. The fifth high priority research area is identifying fundamental principles to produce conceptual foundations for understanding the biological basis of mental processes through developing new theoretical and data analysis tools. The point here is that we have not had the ability to measure neural activity in thousands or tens of thousands or millions of neurons simultaneously. So the fundamental principles are based on what we can measure, which are generally the activity of one neuron or a couple of neurons. Once we get into this new age where we can see brain circuit function at this much deeper level, we are going to need new tools to analyze this data, and it will lead to fundamental new ideas about how brain circuits function. And that's what is meant by identifying fundamental principles. Many of the techniques that are being developed are really only able to be used in animals at this point. And to record from all neurons in a nervous system during a behavior is we are only able to do that in very primitive organisms such as zebrafish, potentially in Drosophila next, and then maybe in the mouse. But the goal of this is to advance human neuroscience. So we can work from the inside out, from the most detailed, controlled situation, and work our way until we understand everything and then move into the human. But the Brain Initiative is also asking us to also start with the human, use the technologies that we have now in more innovative ways, develop improvements in those technologies or new technologies that will give us a window to the human brain, such as the MRI has done in the past. So the Brain Initiative is moving from the inside out and from the outside in. And the focus, the eventual goal, is to create and support integrated human brain research networks and understand them so we can better understand and treat its disorders. And from Brain Initiative to the brain, the idea here is that what we want to do with the technology is to take it to the point where we are learning new things about these important brain functions that are really very difficult for us to measure now, such as cognition, emotion, perception, and action in health and disease. So to take the technology to understand circuit function and its dysfunction. So those are the 7 major points that the Brain's 2025 scientific vision put out for us as goals for scientists in the United States and around the world to reach for, to develop the technologies to be able to put the ball across the goal line and achieve in these 7 areas. The first 5 years is expected to be heavily weighted on technology development because the questions we want to answer need new tools. Once those tools are developed, the plan then is to move more heavily into discovery-driven science using those tools to answer these very important questions. The investment that was recommended in the Brain 2025 report is seen here. The total is seen in black, so it's investment that ramps up over time to approximately $500 million per year by FY19. As mentioned, as seen in blue, the early investments will be heavily in tool development and neurotechnology. And then over time, more of the investment should go into actual use of those tools to answer neuroscience problems. And this needs to be supported by infrastructure for tool dissemination, for data sharing, and for moving the entire scientific field forward. Important to mention, the technologies coming out of the Brain Initiative should be valuable to a broad swath of neuroscience. It's not for particular regions of neuroscience, but for the entire scope of neuroscience that we think can benefit from these tools coming out of the Brain Initiative. Where are we today? In 2014, NIH invested $46 million in 58 projects across 6 different funding opportunities. It funded 100 investigators on these teams in 15 states and 3 countries. In 2015, we're expanding to almost $80 million with 10 different funding opportunities in the areas that were mentioned previously as outlined in the Brain 2025 report. The hope is that we will receive what was proposed in the President's budget, $135 million in FY16, to get us closer to our targets. So in summary, brain disorders are a leading source of disease burden and cost in the U.S. and around the world. Recent breakthroughs and new technologies are transforming how we study brain structure and function. Yet, we are still lacking the tools we need to be able to monitor and modulate, intervene in brain circuits for the benefit of health. The Brain Initiative builds on the recent progress to create tools that will accelerate discovery and build the foundation we need to reduce the burden of brain disorders. I hope that this was informative, that intrigues your interest into the Brain Initiative, into how the brain functions, into the ability of new tools to open windows into the brain, and to use those tools to understand brain disorders and how better to develop treatments for them. I'd be happy to answer questions at this time. Thanks very much for your attention.
Thank you, Dr. Koroshetz, for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is the Brain Initiative currently focusing only on understanding normal circuit function, or are you looking into diseased brain at the same time?
Good question. The NIH Brain Initiative is concentrating in both of those areas in truth. But at this early stage, the emphasis is on tool development, technology development. There are, however, projects that we hope to launch very quickly in which new technologies can be used in patients undergoing neurosurgical procedures, such as deep brain stimulation for Parkinson's disease, or intracranial EEG monitoring for persons with epilepsy, to better collect data on brain activity in those regions where neurosurgeons have put devices and put hopefully new devices that are coming out of the Brain Initiative to learn better what is the abnormal circuit function based on the brain activity measurements that they're making, and also how better to intervene with brain stimulation. So for instance, we have projects which bring companies that develop cutting-edge new technologies in these neurosurgical areas will be working with our scientists to better understand brain function and how to improve it in people with these disorders. I hope that answers the question. Thanks very much.
Please give us some idea of the tools being used or developed currently, ? For example, molecular biology or imaging or electrophysiology or bioinformatics, etc.
Yes, well, there are a large number of tools that have come into the foray, say, in the last 5 years, and I'm going to be remiss in not mentioning them all. I did mention optogenetics, and that is incredibly useful now in studying what happens when certain nuclei in the brain are turned on or turned off. That involves placing optical catheters into the brain, and this means animals, to turn on a light that will then activate the channels that have been introduced into the nuclei of interest that either turn them on or turn them off. There is another technology called dread technology. That's extremely interesting, somewhat parallel to the optogenetics. But there a gene has been introduced with a receptor, and that receptor does not activate with any known neurotransmitter, but instead activates artificially when the animal is given a drug. The drug that's been used mostly so far is clozapine. It's a clozapine analog called CNO. In the rodent, it's very inert. In the human, it actually does metabolize the clozapine, which is an antipsychotic, so it has actual biologic effects. But recently, through the Brain Initiative, investigator Brian Roth's group in North Carolina has developed the ability to activate this receptor with a drug that is inert. And as mentioned, by giving the drug, then the neurons in which this artificial receptor have been introduced can be either turned on or turned off, giving us control not necessarily from an optical catheter on the inside of the brain, but from a drug that can be given intravenously or orally. So that's an example of movement of the technology in this area. In terms of brain recording techniques, I mentioned deep brain stimulation, where wires are placed into the brain, currents injected in patients with Parkinson's disease. These wires are now being developed to have recording capabilities as well as stimulating capabilities. This will enable continuous data to be obtained from people who undergo this therapeutic procedure. So we'll have real data on brain activity in this region where the wire is, and that can be correlated with behavior and correlated with the current injection, the intervention that's made. There are also great advances that have been made in the brain-computer interface world, where very tiny chips have been introduced in the brain, enabling recording from one or a few neurons per projection, but these chips can have hundreds of projections, enabling recording from hundreds to thousands of neurons. The power of this has certainly been seen in cases in which this brain electrical activity has been used to control computers or prosthetic devices. What we've seen here is that the neural activity coming out of the brain is a mirror on how the brain learns, because when first put in, the neural activity cannot control the device or the computer, but the brain learns how to do that over time. So by collecting this information, we see brain plasticity in real time. So these are just a couple of examples that are new that we could add on to the MRI scanner, the high-field MR, functional magnetic resonance imaging, magnetoencephalography, intracranial ECOG recording of neural activity. So yes, there have been many advances, and that's why we believe the time is right because these are stepping stones to something even more miraculous. So thanks for that question.
Reports are DBS is working for Huntington's. Question mark. Have you been testing that more if it's helpful?
Well, I think I was actually a neurologist who took care of many Huntington's patients over the years. And I have not been aware of DBS as a very effective treatment for the disability that occurs in Huntington's disease. Huntington's disease, unfortunately, affects large areas of the brain, multiple different networks and connections. There is a motor control disorder that's similar to Parkinson's with slowness, incoordination. There's chorea, which is an involuntary movement. But the biggest problem is actually motor planning. So patients with Huntington's disease have great difficulty learning motor control programs to play the piano, wash the dishes, and they lose these motor programs over time. So the problem in Huntington's is very complicated throughout multiple circuits and would be very difficult to turn that around by stimulating one brain area. I think where deep brain stimulation has shown a lot of promise, but still in the investigative stage, is in depression. So patients with depression that are resistant to medications have been helped by turning off a particular brain region with deep brain stimulation. And so these disorders where there's a central control region that is spreading out into large areas of brain, such as the dopamine cells in the nigra, which then release widely into the striatum, is an example where stimulation looks promising. In depression, similarly, there may be certain control centers where changing the activity in that one center can have a massive effect. But in things like Alzheimer's and Huntington's disease, the disorder is really quite distributed throughout the brain. Thanks very much for that question, though.
Which are the companies that are helping in tool development?
Another good question. I hope they all will. It's actually something we're actively working on, is engaging industry in the Brain Initiative. So we've had discussions with companies as far afield as brain imaging companies, to the companies that develop intracranial recording and stimulation technologies, to the companies that are involved in managing and analyzing the huge streams of data that come out of the brain when one measures brain activity in thousands of neurons simultaneously. So we think that there is a great interest on the part of private industry to become involved in the Brain Initiative. But I think it's a work in progress aligning the different agendas in the private industry groups, in the public groups. And for certain areas of the Brain Initiative, they're more relevant to industry than others. I think we've had the greatest pickup in the area of brain stimulation and brain recording, intracranial brain stimulation recording in humans. So thanks for that question.
Will brain initiatives be interested in linking brain function disorder to environmental exposure?
Well, I think looking into the brain and understanding brain circuits is the current frontier. The next frontier, I think, is what this questioner is getting at, and that is understanding environmental influences on brain function. And that has been very difficult. As mentioned, our tools for interrogating and monitoring brain circuits are quite poor. Our tools for monitoring environmental exposure over time in humans are worse. So I think that's another area of discovery and tool development that's crucially needed. So for instance, a disease like Parkinson's disease. There is a genetic component, but it accounts for a minority of the percentage of causality in Parkinson's disease. So that means to us that there is an environmental component, but how to get at that? Certainly, there are very tantalizing natural experiments that happened. The most prominent was the use of a drug called MPTP by drug abusers in California. They were trying to make a recreational drug and instead made a chemical that destroyed the neurons in the substantia nigra and caused severe Parkinsonism. A dramatic example of how an environmental exposure leads to brain diseases. In Parkinson's disease, the question has been for a long time, are there other less potent but longer exposures to toxins that lead to the disease? There are some clues that pesticides may be in that category, but we have a real problem in interrogating an integral of the exposure to toxins to understand how they work to disturb neural circuits. So I think coming together in the end will be our understanding of genomics, our understanding of brain circuit function, and then bringing in the environmental exposures to see how they interact in this triad that determines who we are and how our brains function. So thanks again. That was a great question.
Unfortunately, we are out of time. I want to thank the audience for their interesting questions and participation in today's event. I'd also like to thank Dr. Koroshetz for his informative presentation. Today's webcast will be available for on-demand viewing for several months following. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.